This lecture is going to be about the blood. As you may remember from early in the semester, the blood is classified as a connective tissue. So if we look at whole blood, if you took a tube of blood out of somebody, there's two main parts to it. There's the plasma, which is the water portion of it that makes it blood a fluid, and you can see that here in kind of in yellow, yellowish white. And then everything else are the cells. The cells are called formed elements, and there's three types. There's erythrocytes, which are also called red blood cells. There's leukocytes, which are also called white blood cells. And there's thrombocytes, which are also called platelets. And we'll discuss each of their functions in the upcoming lecture. So I want you to take a quick look at this picture. So basically what they're showing here is that if you took blood out of someone's arm or a blood vessel, you put it in a centrifuge to spin it down, all the cells tend to move towards the end because they're heavier, and then it separates. So the plasma separates from the cells. And looking at this picture, you see that everything that's red is the erythrocytes, and then this little tiny layer that looks kind of purplish, which is called a buffy coat, that is made up of the leukocytes and platelets. So most of the formed elements are erythrocytes. But if you look at the percentage of what makes up the bulk of the blood, it's actually plasma. So plasma makes up on average 55%, maybe 60% sometimes of someone's blood. So here's kind of a breakdown of the things that you find in blood. So the plasma is mostly water, and it has other things in it like some proteins, sodium, potassium, that's the electrolytes, other nutrients like carbohydrates, fats, those would be floating around in there too. Then we have the erythrocytes, and just to give you an idea how many we have, 4.2 to 6.2 million per cubic millimeter of blood. So a cubic millimeter of blood is a tiny little bit of blood, like almost a drop. And so if you could imagine that many red blood cells within that tiny bit of blood, we have a lot when you look at the blood as a whole. For the leukocytes, 5 to 10,000 per cubic millimeter of blood, which still is a lot, but is not a lot compared to the red blood cells. And you can see there's different types of leukocytes, and each of these different types has a specific function. We'll talk about that throughout the lecture also. But for the most part, the function of the leukocytes is going to be an immune function. And then platelets, we have a range here, 12 to 300,000 per cubic millimeter of blood. Still small compared to the number of erythrocytes we have. But for platelets, their main function is to clot the blood. All right, so function of blood as a whole, transportation, so transports oxygen, carbon dioxide, nutrients, hormones like you learned about in the endocrine system. Regulation, the blood helps regulate things such as body temperature, can regulate pH also. And then protection, and that comes in with the clotting aspect and also the white blood cells and their immune function. So starting with the plasma, if we look at it in detail, there's not a whole lot to say other than it's mostly water. It has some proteins and ions in it like sodium, potassium, may have glucose, fats in it like we said. A term, if you were to take all the proteins out of plasma, they call it serum. And then some of the major proteins you have in plasma include a group called albumins, which are very popular. They have a variety of roles, and it's all related to carrying things. Like they carry some hormones, like steroid hormones, through the blood. They carry fats through the blood. And then other types of proteins, globulins, fibrinogen, which plays a role in the clotting functions, and then other regulatory proteins. But out of these, albumins are probably the main one that you're going to see a lot of. So we're going to go in some detail into the red blood cells now. Urethrocytes, and I'll just kind of put all this up so you can see it, and we'll run through this list, not necessarily in order. So this is a red blood cell or a erythrocyte. If you look at blood through a microscope, they'll look like these little pink discs. So they have what is called a biconcave disc structure, and it kind of looks like a donut in some level, with like a jelly donut where the center is still kind of in the middle, or a lifesaver with the center still in the middle. But that's the biconcave structure. They don't have a nucleus, so in development the nucleus leaves. So that makes them kind of unique. Their main job is to carry oxygen. They also carry a little bit of carbon dioxide, which a lot of people don't think about, but it's very minimal. But for the most part, their number one job is to carry oxygen. They do that because they have a protein called hemoglobin. So hemoglobin is a protein in red blood cells that carries oxygen. And I'll show you the structure of hemoglobin in the upcoming slide. Each red blood cell has about 280 million molecules of hemoglobin. So we have a huge ability to carry oxygen through the body because we have so many red blood cells and so much hemoglobin per red blood cell. What I mean here by bright red versus dark red, it has to do with the oxygenation of the blood. So when red blood cells are fully oxygenated, they're saturated with oxygen, they turn bright red. When they're desaturated and they're not full of oxygen, they're more dark red. So you can actually see this sometimes in clinical situations. If a vein which is more common, damaged and someone starts bleeding, the blood seems to be a little bit darker because systemic veins anyway, carry deoxygenated blood. They have some oxygen, but it's not fully oxygenated. If you get a serious injury and it goes down deeper and hits an artery, which systemic arteries carry oxygenated blood, which we'll learn all about this in the blood vessel lecture, then the blood tends to be more bright red. And that's usually a more serious injury because it usually means it's a deeper injury. Other things on the slide, this term hematocrit, you should know what it is. Hematocrit is basically the percentage of formed elements in the blood. And so remember, there's 3 formed elements, but the bulk of those formed elements are the erythrocytes. So the percentage of formed elements in the blood is hematocrit, and the bulk of that is determined by the red blood cells. So based on the percentages that I showed you in the first few slides we went over, if plasma makes up 55% of the blood, that means the hematocrit is 45%. So that's the other percent of cells other than plasma. This is a good variable. It's a clinical variable that's commonly measured just to make sure that you have enough blood cells. And that could be a problem if hematocrit gets too low. It could impair your ability to carry oxygen in the blood. So this is hemoglobin that I was talking about. So it's a big protein. It has 4 chains. The chains are called globins. And each of those chains has this little red dot. That red dot is called the heme unit. And the heme unit, the little red dot, contains iron. Iron is where oxygen binds. So if this is one hemoglobin molecule, one hemoglobin molecule can carry 4 molecules of oxygen. And remember, each red blood cell is going to have about 280 million of these hemoglobin molecules. And we have 4.2 to 6.2 million red blood cells per cubic millimeter of blood. So if you kind of put all this together, you can see that we have an amazing ability to carry oxygen. And that's why we can stay alive and live for so long and get oxygen to all the tissues that need it in the body. So just really quick about the erythrocyte life cycle. A given red blood cell will only live about 120 days, and then it's broken down in the liver and spleen. And all this picture is really showing is that the bulk of red blood cells are recycled. So once the red blood cells are broken down, they are recycled and used to form new blood cells and other things in the body. All right, so that's it for the red blood cells. Let's move on to the leukocytes, the white blood cells. So white blood cells, they do have a nucleus. They don't have hemoglobin, so they don't carry oxygen. There's 2 classes. They're classified as either granulocytes or agranulocytes. So granulocytes are called granulocytes because they have visible granules in their cytoplasm. So it looks like this. Agranulocytes look more smooth, where you don't really see any granules. So there are 3 granulocytes and 2 agranulocytes. And so starting with the granulocytes, most common white blood cell that there is the neutrophil. Neutrophils are identified by having a multi-lobed nucleus, kind of like this. See multiple lobes in the nucleus. You should see the granules in the background. And they make up the most of the cells, so 60 to 70% of the white blood cells. And they basically have a general response. They respond to any infections and they phagocytize bacteria. Eosinophils make up 2 to 4% of white blood cells. This is probably the best picture of one, because they're supposed to be identified by having a red or orange cytoplasm. So red or orange granules in their cytoplasm, so the cell should appear like a slightly different color, like reddish or orange. These have a parasitic function, which kind of makes them unique, and they also respond to any allergic response. So for identification purposes, the reddish or orange cytoplasm with the granules is what you're looking for. So parasitic response, allergic response. And then the last granulocytes are called basophils. So they're pretty big cells. They have these big purple granules in them. They also respond to allergic response and inflammation. What makes these basophils unique is they secrete these substances. Histamine, which causes vasodilation, or blood vessels to dilate and get bigger, and also heparin, which inhibits clotting. So heparin is kind of like a blood thinner. It makes the blood flow more easily. So if you envision if you get inflammation, you think the good way to remember this, if you get an inflammatory response to something in the body, it usually appears red and swollen. Well, the redness comes from increased blood flow to that area, which is due to the blood thinning and dilating the blood vessels so blood can get there. All right, and the 2 agranulocytes, the first one is called monocytes. These are really easy to identify. They have a big C-shaped nucleus, and these are big cells. They're generally much bigger than the other white blood cells. And their function, they turn into macrophages and they go around phagocytizing things. So big C-shaped nucleus. And then the last one, which is really complex, lymphocytes, makes up 20 to 25% of the white blood cells. These are small and they're identifiable because they're relatively small, but their nucleus is big relative to the size of the cell. So the nucleus, in most cases, almost takes up the entire cell, and it just looks nice and smooth. So that's what you should know for identification purposes. And then something we're going to talk about with the immune system is the different types of lymphocytes. So we have T, B, and we also have natural killer cells, which are a type of lymphocyte. So more on this later, but to be really brief, T lymphocytes generally attack foreign and virus-infected cells. B lymphocytes differentiate into plasma cells, which you may remember from early in the semester, which secrete antibodies. We'll talk more about that with the immune system. The natural killer cells just kind of go after anything that is infected or abnormal. So if we try to identify these different leukocytes, I'm just going to kind of run through them so you can see them. And these would be good lab test questions. So this one, you notice what's different about it, its color. So that would be an eosinophil. This one, look at that spiky appearance, big purple granules, kind of like little spikes all over it. That's a basophil. Big cell right here, C-shaped nucleus. That's a monocyte. Here's a relatively small cell with a nucleus that takes up most of the cell. Looks smooth. That's a lymphocyte. This one and this one are general-looking cells with multi-lobe nuclei. So these would be neutrophils. Neutrophil, neutrophil. All these pink things in the background would be red blood cells. All right, lastly, thrombocytes. So thrombocytes are platelets, same thing. Their job is to clot blood. So they're an interesting thing about the thrombocytes is they're not true cells. So what happens is you have a cell called a megakaryocyte, looks like this, and then it just kind of breaks apart and it fragments. and all the little fragments are the thrombocytes. They're platelets. So it's kind of, when you say thrombocytes, site means cell, it's actually not completely correct because they're not true cells. They're like fragments of cells. So they don't have a nuclei, they're not true cells, but they form blood clots. They prevent you from bleeding too much, and they make the blood clot. All right, so we're going to do a little bit on blood typing. And so there's a lot of different blood typing systems, but in general, they're based on the presence or absence of what we call antigens. So antigens in this case are proteins on the surface of red blood cells. So the two most common systems that are used, and this is what we use in the United States, there's the ABO and RH system. We use them together. So in other words, your blood type could be A, it could be B, it could be O, or it could be AB. And that's also going to have a positive or a negative with it. The positive or negative refers to what we call as the RH. You're either RH positive or RH negative. So if your blood type is O positive, that means you have O type blood and you're RH positive. All right, so to try to explain what I was getting at with these antigens, so if you look at these red blood cells down here, you see all these little kind of proteins on the surface. Those are called antigens. So these antigens is a term that's commonly used to refer to things that are bad in the body. Like if something gets into the body that can be harmful, like a virus, they call it an antigen. In this case, these are not bad. So in this case, we're just talking about surface antigens on the surface of red blood cells. So in this cell, it has antigen A. And it looks like they're just blue. That's just fine. This red blood cell has antigen B. This one has both. And this one has none. Okay, that's the basis for our ABO system. So if your red blood cells only have antigen A, then your blood type is A. If your red blood cells only have antigen B, you have blood type B. If they have both A and B, you're type AB. If you don't have either of those, you're type O. That's as simple as it is. And so that's what determines your whether you're A, B, or O, or AB. Now, the confusing part of this is when you start talking about antibodies. So just keep in mind, antigens that we're talking about now are on the surface of the red blood cell. Antibodies could be specific for a given antigen. So long story short, if an antigen is specific for an antibody and they come into contact with each other, they're going to bind to each other, and something in the blood is going to happen called agglutination. You don't want that. It's going to be basically causing like a big mess in the blood. So in other words, if you had antigen A on your red blood cells, you would not want antibody A in the plasma because they would interact and cause problems. So keep that in mind, and now we're going to talk about the antibodies, and I'll keep trying to keep those separate as two different things. Antigens are on the surface, antibodies are in the plasma. So the plasma contains antibodies. Antibodies attack antigens. So when antibodies attach to antigens, agglutination or clumping of the blood occurs. Remember, you don't want that. So in type A blood, remember it had surface antigen A, so it can have antibody B in the plasma because they won't interact. They're not specific for each other. In return, type B blood, which has antigen B on the surface, can have antibody A in the plasma, and that's how it is. And then type AB blood, remember, had both antigens, A and B antigens, so it can't have any of those antibodies in the plasma, because if it did, agglutination would occur. Then type O blood, which has no surface antigens, you can have both antibodies in the plasma, A and B. And so this is where most people get confused with this, but if you keep it separate, I think it won't be that bad. So if you're type A, just think about this. If you're type A blood, that means you have surface antigen A. You do not want antibody A in the plasma because they're gonna react and cause trouble. But you can have antibody B, and that's what they have. All right. So hopefully that makes sense. So this hopefully will drive this home. So type A blood, surface antigen A, it has B antibodies in the plasma, everything's good. Blood type B has surface antigen B, A antibodies in the plasma, everything's good. Type AB blood has surface antigens A and B, no antibodies. And then O, no antigens, both antibodies. All right, so with that being said, that allows us to type people's blood using this system. So this is a common lab that we would do, and I'm basically going to talk you through it right now. But when you type someone's blood, it's based on this agglutination or clumping of the cells. So to tell whether it's A, B, O, or AB, we get a vial of antibodies. We have a vial of antibody A, a vial of antibody B. We take 2 drops of blood for starters. We put 2 drops of blood on a piece of cardboard or a piece of paper, and we put antibody A on one drop. We put antibody B on the other drop, and we just sit back and watch what happens. So in this case, only the drop of blood with antibody A on it clumped and agglutinated. The one when we put antibody B on it, nothing happened. And so what does that tell us? That means antigen A is present in this blood because it bound with the antibody A. So they don't have antigen B because nothing happened when we put antibody B on it. And so this person has type A blood. And then this next example, if only this one clots, that means they do not have antigen A, but they do have antigen B on the surface of their blood cells, and so they're type B blood. If both agglutinate, that means they have both surface antigen A and surface antigen B, that means they're type AB. And if neither agglutinates, that means it's type O, they have no surface antigens. And then similar to test Rh, you just use a different antibody. So it's antibody D, or sometimes they just call it the Rh antibody. And same thing. If it's there, you're Rh positive. If it's not, you're Rh negative. So same thing. You just take this other vial of the Rh antibody, put it on a drop of blood. If it agglutinates, this is showing someone who is Rh positive. This is showing someone who's Rh negative. And it's that simple. All right, so last thing, blood types. As far as the prevalence, you can kind of look at this, and this is in the United States. Based on the percentages, most people in the U.S. are O positive. Next in line would be A positive. Then after that, it drops off pretty significantly. You see B positive at 8%, and then you start running into things like O negative, A negative, rarest AB negative. So generally when we do this in class, most of the things that we see are O and A. All right, and lastly, as far as who can donate blood to who, universal donor is type O because it has no surface antigens, so any blood type can receive it. The universal recipient is type AB because they have no plasma antibodies, so they can receive any type of blood. Doesn't matter what antigen it has. And then if we want to be more specific, Rh negative can only receive Rh negative blood, whereas Rh positive can receive either positive or negative. And it has to do with the lack of the antibodies in the blood. And here's a more specific chart down here, but for the most part, if you kind of know this up here, you should be good. All right, now this is really the last thing. Blood cell formation. So formation of blood cells is called hemopoiesis, sometimes called hematopoiesis, same thing. But the key thing that I want to get across here is that all blood cells, here's the ones we just talked about, your erythrocytes, platelets, all your white blood cells, they all come from a common stem cell. So this stem cell is called a hemocytoblast, common stem cell. It's going to differentiate into what we call a myeloid stem cell or a lymphoid stem cell. So from here, the lymphoid stem cell gives us all our lymphocytes, B lymphocyte, T lymphocyte. The myeloid stem cell gives everything else. So I don't really want you to know all these little steps in here, but what I do want you to, I want to point out a few things. One, you can see in the red blood cell formation that the nucleus gets ejected in the formation, and so that's why it doesn't have a nucleus. In the platelet formation, you see you get a megakaryocyte, megakaryoblast, megakaryocyte, and then it breaks apart and you get the platelets. And then for the white blood cells, really, you don't really need to know any of these steps, but I'm going to give you one more little bit of information, the hormones involved in this. So the hormone involved in red blood cell formation is erythropoietin, stimulates red blood cells. Remember, that comes from the kidneys. Thrombopoietin is a hormone that promotes platelet formation. And then for the white blood cells, they're just called colony stimulating factors. And there's different ones.